today we'll be really talking about how micro-C really breaks this high-C resolution barrier and really what that means for you as an end user of the product, what it can help you find, and then we'll dive kind of into the applications of that as well. And just to reiterate, I'm Corey Padilla. I kind of head up our scientific affairs here, kind of do a little bit from helping in developing products to actually developing some computational pipelines to look at these data as well. So I kind of span the spectrum of these products. So feel free to reach out with any questions and we'll address those during the course or at the end of the webinar. So to get started, I just want to give you a bit of an outline into what it is we're going to be talking about today. So we'll do a very brief introduction on chromatin conformation, why it matters, and how people conduct 3C assays. And then we're going to dive into the limitations of those current approaches to high C, mainly being the restriction enzyme. And then we'll do a deep dive into micro-C and how the MNase really helps break this resolution barrier that's set out with restriction enzymes and current high-C approaches. And then we'll talk about further leveraging micro-C in epigenetics, and mainly this is HiChIP. So we'll give you a little bit of flavor of how you can do protein-directed chromatin conformation with the MNase-based approach to high-C. So let's talk about chromatin conformation. So to set the stage here, many epigenetic assays capture features in linear space, right? These are heavily rooted in primary order epigenetic features such as methylation, histone mods, and chromatin accessibility, right? We know these things as bisulfite sequencing, Chip-seg, either targeting histone modifications or DNA protein interactions such as CTCF or NANOG, things like that. And then ATAC-seq, where you're asking where is transposase accessible to cut and tag DNA where basically nucleosomes aren't. The thing that these sort of primary feature based assays really have in common is that they capture things in linear space, right? So typically what you see here is these data are often plotted out as, your position on the genome and the x-axis, and on the y is your enrichment or depletion of that signal, right? So here we can see methylation drops here on CpG. For Chip-seg here, we see where the proteins are binding to or anchoring to the DNA. And then ATAC-seq, we can see where transposase again can cut and tag, and you get enrichment over those accessible regions of chromatin. But what we do know, and it really over the past 20 years or so through 3C technologies, is that genomes aren't organized like this. They don't come packaged in a straight line, right? In the human genome, each cell has to pack about 3 meters of DNA into a nucleus. And so that comes with this hierarchical folding of the DNA, and that builds up some of these features that we can detect in chromatin conformation. And so some of these you're looking at active compartments that you're euchromatin or heterochromatin and B compartments, where you get more open chromatin that's transcriptionally active, sort of has a tendency to cluster next to other active chromatin, and heterochromatin has a tendency to cluster next to other closed chromatin. Regardless of their distance in linear space, their physical space actually is quite close. Then we have these topologically associated domains, or lovingly called TADs, where these are often CTCF cohesin-mediated events that kind of segregate linked function genes into these topological boundaries. And then you can zoom in even further and look at these gene loops where you can start asking questions about the transcriptional machinery, such as your enhancers and how they're interacting with promoters. And today we'll show you how you can even overcome or zoom in past these gene loops and really start asking questions about enhancer promoter interactions in a very high resolution way. And so really, the question is okay, that's cool. 3D genomics is a thing, but how does it help us? And the answer to that is it really helps us understand the transcriptional landscape of our genomes, right? And so, like I was pointing out, the epigenetics tools out there often give us this linear view where you have your transcription factors and your acetylation marks and methylation marks, those post-translational modifications, right, kind of spread out in linear space. So you get this very linear view. And we know that's actually not how that works, right? So like I was just saying, we know that these enhancers and promoters interact with each other, and the distance, the linear distance that these interactions span are on the order of tens to hundreds of kilobases, right? And so what it allows us to do is really start asking questions about which enhancer and what transcription factors are actually interacting with target promoters. So we can actually start getting a more complete view of how transcription is mediated and kicked off in different cell types or different regions of the genome. And so how do we capture this? How do we go from this linear view to this, three-dimensional folding view? And the answer is with chromatin conformation capture. Now, this technology has been around for about 20 years, meaning this core workflow. And all the different flavors of HiC, your capture C, your HiChIPs, your Chia pets, your 4C, 3C, they all start with this same core workflow where chromatin is cross-linked. That chromatin is then digested. Historically, that's been done with restriction enzymes, and we'll show you that's maybe not the best idea. It was great in the early days. It gave you a very nice anchor point to move forward, but the technology has evolved quite a bit over that time. And so once that chromatin is digested, then we have a ligation event occur. Now, these ligation events are about generating chimeric molecules that are representative of 3D space. So your ligation is likely going to capture something that's close in physical space rather than in linear space. And then we reverse those crosslinks, and what we're left with is chimeric DNA. Again, that's reflective of what's close in physical proximity to those cut sites. And then from there, this chimeric DNA, you could do a smattering of molecular biology approaches to ask particular questions. So back in the day, you would do something like a qPCR where you were asking questions about two specific loci, and you wanted to quantify how often those interactions occur. That was 3C. With 4C, kind of that you're looking for this blue molecule, and you want to ask the questions of what interactions are associated with this blue loci. So you take that chimera, you turn it into a circle, do inverse PCR. So you have flanking regions of blue, and so then you can ask questions of the red. Then you could do a capture approach where you have a set of loci that you're interested in. So you build up oligos to target those many loci, and then you pull down and you can ask, what is interacting with my known target region. So this moves from like a one versus all scenario to a many versus many. You're capturing many loci, and you're asking what are the other many loci that are interacting with them. The world has largely transitioned into HiC, which is you biotinylate that chimeric event. You pull down, make a sequencing library, and then all of your chimeric molecules are sequenced, and the reads are informative of that 3D space. Then you can do protein-assisted approaches where you come in and you pull down with an antibody of interest against protein of interest. And then you can either do a qPCR approach, or you can combine that with a sequencing-based approach. And so it's kind of like combining your Chip-seq with HiC. And so those are the many different flavors. The first part of this, we'll talk heavily about HiC, and then we'll close out with a little bit of the protein-assisted version. And the way these data look are basically based on read pairs mapping to the genome, right? So here we have each one of these dots here represents a read pair, and on our x-axis we have genomic position, but on our y-axis we have read pair separation. And so each one of these dots again represents a read pair where read 1 maps to here and read 2 maps to here. And as we get closer, we can start seeing that there are more contacts as that the distance becomes smaller. What this looks like in real life, though, is you get these sort of triangle features, or if you're looking at it in a pairwise fashion, square features of regions that are highly interacting with each other. So these sort of triangles represent regions that interact very frequently with each other, and then these red dots indicates a much higher degree of interaction. And so we can really start looking at what these high interaction frequencies regions encompass. Are they enhancer promoters? Are they CTCFs that are interacting? And so we can really dive in to start figuring out what is interacting with what. So basically teasing apart the interactome of the genome. So now we're up to speed on HiChIP and how the data look. And now we're going to dive a bit into the limitations of current HiChIP approaches, and mainly this revolves around the use of restriction enzymes. I have seen a few questions pop up. We'll save those till the end, just so we can get through the slides, and then maybe I'll address a question along the way with the slides that I already have. If not, then we'll come back and address that. So the fundamental limitation of restriction enzyme-based HiChIP is resolution. And so this is a phenomenal review paper, Davies et al. 2017, that really outlines, what are the applications, the usabilities, and the different flavors of proximity ligation. And this image really pops out to us, because it's talking about resolution. And so there's a lot of definitions you could put around resolution, but the very basic one is how far can you zoom in on chromatin conformation features and have enough read support to actually have confidence, statistical confidence that what you're seeing is a true interaction, right? And this resolution is very much based upon two things. It's your functional resolution, which is your fragment size, and your contact matrix resolution, which is your read support per fragment. And so if we look at the functional resolution, which is your fragment size, if you approach with a 6 cutter restriction enzyme, your fragment length is going to be highly variable. Now we can shorten that fragment length with a 4 cutter to kind of tighten that window and makes for smaller fragments, but you still have a wide variance here in your fragment size. When you use MNase to chop up the chromatin, you get nucleosomes, right? You get, and that is a very nice titer around that nucleosome. So you get fragments that are around 146 bases. Now, when we move into resolution, right, which is how do you have to bend your genome and able to look at it in a contact matrix? And I'll show you a bit more about that bending here soon. But we could see that the larger fragment size really puts a limit on your resolution. You have to generate these 40 kb bends. And then when you start looking at 4 cutters, you can do 5 kb bends to 1 kb bends, but that's really functioning on your read depth, right? How much reads do you have to support this? What is interesting is that MNase is the only approach here that maintains the function, or the contact matrix resolution is the same as its functional resolution, and I'll show you why that is here in a few slides. Another limitation that really actually adds a lot of burden to your sequencing depth is your genomic coverage. So restriction enzymes really introduce bias into your genomic coverage because you're enriching data over restriction sites, right? And so what we see is if we ask a question about genomic coverage so number of bases that exhibit different coverages, in the dotted line, we have shotgun data, just normal whole genome sequencing data, so you get that nice Gaussian distribution. But when we look at the single restriction enzyme HiChIP data, we see a lot of the genome that exhibits low to no coverage. And on the other side of that, we see quite a bit of the genome that is overrepresented in sequence space. And these are the regions that contain high density of restriction site motifs. And so this is a very inefficient coverage. So if you wanted to sequence more of your genome, it's a lot more sequencing to move this mean over. This is going to get sequenced quite a bit more, but you're still never really going to bring these regions along for the ride. With a multiple restriction enzyme approach, it does in fact help that coverage distribution, but it's still dependent on restriction sites. So you're enriching data again over restriction sites. It's a little bit more diluted because there's more restriction sites, but you're still leaving behind regions of the genome that are sort of restriction enzyme deserts. So that coverage really makes sequencing Hi-C data kind of inefficient. And we'll address that here in a few slides with micro-C. And the last real limitation of Hi-C data is the types of molecules that are generated during the ligation. So here, if we just walk through what the Hi-C reaction is again, so we have fixed chromatin, and here I'm indicating where the restriction sites are. And then we digest that chromatin with restriction sites, so we get these fragments, and then we conduct ligation. Now, there are three main possible ligation events that can occur. So you get a self-ligated event where this right here forms a circle. You get a religation event where you cut and just simply religate back to your original cut site, or you get this valid ligation where the physical distance that's being spanned by this ligation event is larger than your library fragment, right? So the self-ligation and the religation, the insert size represented by this black bar here is shorter than your library fragment. So you're essentially just sequencing whole genome sequence data. You're sequencing shotgun data that's not informative over 3D space. When your ligation event is longer than is the physical distance in your ligation event is bigger than your molecule that you're sequencing, that indicates that you are actually capturing that chromatin conformation, those long-range informations. Now, to convert this into a library, you shear, right? Because you need to be able to add on your sequencing adapters. So when you shear, you turn these circularization events into sequenceable or taggable molecules for library generation. So now you have a lot of data types that actually aren't informative over chromatin conformation. And these typically make up anywhere from 15 to 25% of a Hi-C library, with 75% being these valid Hi-C interactions. And so now we're going to break down each one of those sort of barriers into or limitations in Hi-C and how micro-C addresses those. So micro-C is Hi-C. It is at a very fundamental level. We start with cross-linked chromatin. And then the big difference here is during fragmentation, we introduce MNase to do the fragmentation rather than a restriction enzyme or a cocktail of restriction enzymes. Then we do ligation, where that ligation is mediated by a biotinylated bridge. We reverse the crosslinks, giving us chimeric DNA that's reflective over 3D space, and then we sequence. So no difference, except for the MNase. And so what this means for us as end users is that it gives us resolution down to the nucleosome. So here's a schematic. I'll follow this up with real data, just so everybody can kind of get a picture of why we can build models like this. So here we have chromatin with DNA turning around nucleosomes. Here I'm flagging restriction sites. And then here's a promoter regions where nucleosomes typically don't occupy. And so what we get are fragments. Here are the digestion fragment that are very consistent and uniform in size, right? So we get these nice fragments that are all about the same size compared to restriction enzymes where the size is very variable and dependent on the distance between the two restriction sites. Now we can ask the question, what is the ligation links between these? So you can start capturing all of these subtle ligation events that occur at the nucleosome scale, which really the ligation events you're capturing with Hi-C are really reflective of your restriction fragment, right? That's really the ligation events that you can see. And then when it comes to coverage, we build these nice histograms over each one of the nucleosomes, right? That's, we're getting nice uniform coverage over each one of these nucleosomes. Whereas with restriction enzyme-based HiC, since you biotenylate at the cut site in the ligation event, you're actually piling reads right at the cut site. And so you have to split this read support per each one of these fragments. So yeah, you get, a nice enrichment and coverage right there, but it's half of this is only informative of this fragment. The other half is only informative of that fragment. So you're splitting your read support per the ends of your fragment rather than enriching over the nucleosome. Okay, that's cool. That's a schematic, but what does that actually look like? So here what we're doing is we're taking micro-C data and we're plotting it in, just as coverage, not looking at any of the proximity ligation. We're just like basically looking at your BAM in IGV. And so we're doing a metagene analysis around high occupancy CTCF sites with micro-C, multiple restriction enzyme HiC, and single restriction enzyme HiC. And as you can see, you get this nice oscillation where the peaks are nucleosome protected DNA and the valleys are linker DNA that are digested during the MNase fragmentation. Now, you don't see that pattern with restriction enzymes because you're not enriching over something that is chromatin centric, you're enriching over a primary sequence motif. So it's less informative over the chromatin space than a micro-C assay would be. And so now, if we plot this out, like we would in any one-dimensional sort of epigenetic view of coverage of these features, we can see here that we're positioning nucleosomes quite nicely, where you don't really see that in the multiple or single restriction enzyme. Moreover, we see these big old large gaps in coverage for both the multiple and single restriction enzyme, because these are restriction enzyme deserts. And I'll show you in a few slides what this means in terms of building contact matrices and why even binning these data at 1 kb can be a challenge, where micro-C actually works quite well. So one of the questions that often comes in when we talk about MNase and into chromatin digestion is about what happens over nucleosome-free regions. A lot of people talk about promoters. People bring up attack-seq as, shouldn't you see somewhat, see loss in data where you see an attack-seq peak where nucleosomes don't live. So here what I'm showing on the human genome is chromosome 8. Here's micro-C data across the genome. Here is attack-seq, here is H3K4 trimethyl to indicate, active promoters. Then we have RNA-seq here to just show activity. And so as you can see, we cover the genome really quite well. And we do have some oscillations, that is true, but it's not as pronounced as you would expect over like an attack-seq type assay. And if we zoom in on really any random spot here and ask, what's the coverage looking like where we see an attack-seq peak and an active promoter that's currently being transcribed, we do see that, yes, there are a few dips in coverage, right at that attack-seq peak, but coverage never really quite goes to zero. The reason being is that our digestion profile is a mix of monodi and trinucleosomes. And so while you're still enriching over the nucleosome, that linker DNA is preserved at some very basal level. And so just to show that this, like, we're not really trying to cherry pick regions here, is we compare that genomic coverage in terms of bases exhibiting X amount of coverage compared to whole genome sequencing data, and then compared to restriction enzyme based HiC data. And we can see this nice distribution with the whole genome sequencing data. We're actually able to recapitulate a lot of that normal distribution. It is less effective because we are sacrificing more linker DNA and we are enriching over nucleosomes, but we still get a fairly normal distribution. And when we compare that to restriction enzyme-based data, the coverage is just way, way, way better. And I'll show you why that's important in building contact matrices. So the last kind of bit of proof here to that sort of promoter region coverage or nucleosome-free region coverage is I've built contact matrices on the human genome at 1 kb and at 20 bases. And I simply ask, what is the coverage? How many reads support each bin for all bins, bins that don't overlap promoters, and bins that overlap a promoter. And what we actually see is there's a slight increase of coverage over promoter regions, largely because that when you do see active promoters, those are usually associated with a histone mark, right? H3K4 trimethyl. So there is a histone nearby that we're capturing. And we also have some, evidence to suggest that in nucleosome-free regions, you often have a protein that's bound there, right? That might be POL2, it might be an anog, it might be another transcription factor, but at some level, that protein bound to the DNA helps protect from MNase digestion. So we aren't really losing coverage over these regions. And when we even bin at smaller fragment size or smaller bin sizes, we basically just divide our coverage by 5, which makes sense because there's 5 200 base pair bands and in a 1 kb band. So that all makes sense. And so finally, to kind of get at the last limitation of the restriction enzyme-based Hi-C data are, these types of ligation events and the noise that's introduced during shearing of the self-ligation products, right? So here I'm showing that workflow that I showed earlier compared to a DNase or MNase-based approach. One thing to point out here is that our molecule distribution is so small during fragmentation that we actually don't need to shear to convert these molecules in the library. If you take an extreme, you ligate 2 4 kb regions together from a restriction enzyme digestion, it's an 8 kb piece of molecule that you then have to shear to convert into library. If all your molecules are fairly small, even after they ligate, they're well within the range to just convert into a sequence library. So you don't have to shear, and we don't shear. And so what that leaves us is with short religation events and valid religation events. And to help mitigate these religation events from occurring, we add a secondary crosslinker that has a longer arm than formaldehyde. And so that it basically just builds a more robust matrix or fixation matrix. So we're limiting the amount of short inserts, and we're enriching for these long ligation events. And if the question arises about what is a valid Hi-C read without a restriction site, well, it's pretty much the same definition in that the ligation event has to be longer, has to have a physical distance longer than your molecule that you're sequencing. But now you just don't have to depend on your ligation event being adjacent to a restriction site. Okay. So now that we've talked about the limitations that MicroC overcomes, let's talk about what that means for you as an end user in applications. So just to sort of show here that we are in fact generating more useful data, we're comparing MicroC to multiple and single restriction enzyme approaches, and we're asking of the total library, what reads are valid? And valid reads are reads that have an insert size greater than 1 kb, which is bigger than the library fragment, and trans reads. And so we're getting, between 90 to 95% of our data are valid reads, or like I was saying earlier, with restriction enzymes, between 70 and 75% of the data are valid reads because they have those self-ligated and those religation events occurring at a 15 to 25% frequency. Now, of the cis reads, we're asking what's the insert size. So greater than 1 kb, we're at about 95%. So almost all of our molecules are in fact capturing a distance that's longer than the library fragment. And then if we go to the extreme here of what's longer than a megabase, 50% of our data captures information spanning a megabase. And then in terms of complexity, how many unique molecules are going to get per a Hi-seq lane? And because that genomic coverage is so much more uniform, our complexity is quite higher. So we're getting about 80% unique molecules from these libraries. And so then what does that mean from a contact matrix standpoint? So if we look at a contact matrix at 4 KB resolution at 800 million read pairs across the different approaches to proximity ligation, here we're calling out known loops that were detected in Raudal, and we're just asking how many reads support each one of these events. And across the board, micro-C is capturing way more read support per each one of the events. But more specifically here, there are events that really aren't being captured that well here in the HiChIP data that micro-C is just really able to detect. And so, or is able to visualize. And so if you call loops with these data, you could see that micro-C is covering, almost 56,000 looping events with an overlap consistent with Raudal, what they saw within their replication samples. And so typically what we see here is these high degree of overlapping events. These are CTCF mediated boundaries. But what makes up these sort of larger circles, or this larger part of the Venn diagram, these are enhancer promoter interactions that just really can't be seen with restriction enzyme-based HiChIP data. You can see more of them than you can with the 2014 approach to HiChIP, but micro-C really does a great job of discovery. And so if we just kind of ask, to get this level of high confidence overlap, like just can we capture the CTCF mediated events that you would see in the RAU data set with our HiChIP or their micro-C data, at 800 million read pairs, that improved coverage makes these bends highly more informative. So we're able to recapitulate those high confidence overlaps, those CTCF mediated events at only 800 million read pairs, which you would typically have to sequence to about 1.2 to 1.6 billion read pairs with restriction enzyme-based HiChIP data. So let's talk about why that is. Why is this doable at 800 million read pairs at 1 KB resolution? So if you think back to the image I showed about, yes, we can position nucleosomes, and here is a restriction enzyme desert. These coverage profiles really matter when you're building your contact matrices. So a typical approach to building a matrix is you bin data into fragments and you ask how much coverage, how much read support do I have per my bin to look at these interactions. So with micro-C, you could build bins at nucleosome scale, so about 200 bases. And so if we do that between these two profiles, we see that overall, micro-C has highly supported bins at 200 bases. There are a number of bins that exhibit slightly less than ideal coverage. They're still a bit informative though. But when we compare that to the HiChIP data, there's regions of the genome that just aren't being covered, right? It's almost pointless to bin HiChIP data at that resolution because these regions are just not informative. And so the next approach there is to bin at 1 KB. So if we do that, we see that binning at 1 KB rescues some of these regions with slightly lower coverage into usable bins to do statistical analysis on, whereas with the HiChIP data, this region still is fairly unusable. And these two regions, because they're flanked with low coverage regions, are okay, but they're still not as good as they can be. And we're really left with only one bin with really sufficient read support. And so, your next thing would be build a bigger contact matrix. So if we do that at 4 KB, I'll just show you what the matrix looks like. It's a dramatic difference in the quality of the contact matrix. So again, we can see all of these internal interactions as well as these larger looping features that, again, you're just not really able to support that well with HiChIP data. And it's important to note here that, in order to really use a region like this, you would need to sequence 35 to 50% more to really bolster the read support. But this region's kind of always going to lag behind, right? There's just no data there to support any interactions. And so what that does for us is it gives us a very finite view of of chromatin interactions. And so this is a figure that was modified from a really phenomenal summary manuscript that really just talks about what are the features that you can now detect when you have resolution capabilities that MicroC offers. So here we have our CTCF mediated boundaries that you can detect with HiChIP data, right? And you can see these TAD boundaries, which again, you can see in HiChIP data. And you can see some loop extrusion features, which historically has been able to be captured with HiChIP data as well. But as you start looking closer into the sub-TAD features, which we know are more dynamic in between sample types, you can start defining these enhancer promoter dots and stripes, right? How they're actually interacting and kind of rubbing up against each other. And we can capture these weak pause events during loop extrusion. So these are, you're able to do the very standard higher order chromatin conformation analyses such as AB compartments, TADs, loops, but then you can go beyond that with MicroC and start getting into these enhancer promoter interactions and loop extrusion dynamics. And so here's a real example of that. And so if we look at a MicroC contact matrix, we can see that there is a stripe here, which has largely been denoted as loop extrusion, where you have a cohesion, you have a CTCF that's anchored, and then you have these transient CTCF binding sites where as the DNA is being extruded through that cohesion, it kind of hits that weak pause sites, stops, you see a little hot spot here in contact, and then it moves until it hits its final anchor, generating this TAD boundary. And as we move closer in, we can start seeing promoter-promoter interactions where here are your two promoters, transcription factors mediating some of those interactions, and then POL2 doing its job. And then enhancer promoter dots where you have your enhancer and transcription factor interactions with your promoter of interest, and then you see the POL2 kicking off transcription there. And so one of the last things that we really want to highlight here is that this nucleosome positioning really gives us a very clear view of chromatin dynamics. The use of MNase is very informative over chromatin space, whereas restriction enzyme data are not. And so here we have a metagene analysis around high occupancy CTCF sites with MicroC data versus a multiple restriction enzyme approach. And as you can see, there's a very clear topological boundary here where this region is interacting with each other more than they are across these two regions. You can kind of see the barrier in restriction enzyme-based HiChIP data. So yes, there is a bit of a barrier there. But there's a lot of background noise here associated with that. But what we can actually see here in the MicroC data is here are each one of the nucleosomes, and here is how they're interacting with each other. And what we see is there's very little interaction between nucleosomes spanning CTCF sites, indicating that CTCF is probably a barrier of chromatin interaction here, which again was described in a paper out of Darzak's lab a couple of years ago. So really the idea here is that MicroC is very much chromatin centric for chromatin conformation, whereas restriction enzyme-based HiChIP is really informative over restriction fragments, which aren't necessarily, right, they're not the building block of chromatin. The building block of chromatin is DNA turning around a nucleosome, and MicroC gives us that view of chromatin dynamics. And so one of the other questions we get is, what about the workflow here? How much do we have to change? And the answer is you really don't have to change that much. On our GitHub page, we have some QC scripts that really take you from FASTQ into a valid pairs file. And then we help you build contact matrices. But we're really not doing any magic here. We're using BWA mem with the -5SP flag to generate our BAM. And then we generate our pairs files using pair tools, 4D nucleome standard. And then from there, we use those valid pairs to build contact matrices with either Cooler or GSER. And then you can do, very, very typical analyses. You can do one-dimensional pileups. I use Deep Tools compute matrix. You can position nucleosomes just by looking at the coverage profile. You can call significant interactions. I'm a big fan of FitHiC. There are a number of other tools out there. This is just a smattering of tools that are out there. And then, for your standard topology analyses, there's Juicer, Arrowhead, there's HiChIP, there's Pikachu, there's Mustache, there's SIP. That's another one that works very well. And then AB compartments, Juicer eigenvector, you just plug in your .hic file, and then you can do your two-dimensional pileups with the contact matrix. There's Juicer has APA. Cool Tools has a really good walkthrough on how to do that with cooler files. And then there's a really cool tool that recently just got published called Cool Puppy, where you just feed in your matrix, feed in the bed file of interest, and it'll generate those pileups for you. So again, this is a smattering. It's not comprehensive, but it's really to show that you don't really have to do anything different than you currently would with any HiC workflow. And so to end, I'll quickly discuss, how to leverage micro-C a bit further in epigenetics. And this is, HiChIP, the protein-directed view of chromatin conformation. And so here, we showed this image for micro-C and how it's giving us these nice fragments that are the size of a nucleosome and how we can capture all the ligation events per nucleosome. So now if we think about CHIP and really kind of dialing in our chromatin conformation questions to say enhancer regions or promoter regions, right? We bring in chromatin immunoprecipitation, which is very centric to, in this case, to chromatin dynamics and the nucleosome. So the workflow is simply combining, I say simply, but we've done a very good job here to try to keep this workflow as simple as possible, to go from cross-linked chromatin to fragmenting with MNase, to then introducing your immunoprecipitation with your antibody of interest. Then we conduct proximity ligation, reverse your crosslinks, and sequence. So really the only difference here is we're adding in the IP. And what that gives us is a view of chromatin that captures both the Chip-seg signal and those HiC chromatin interactions. So here's just normal Chip-seg against CTCF. And looks like normal Chip-seg data. We see nice peaks where CTCF is bound to the DNA. Then we're comparing that to our HiChIP, where again we can recapitulate the binding site enrichment. But now we can ask what's interacting, which CTC factors are interacting with each other, and we can see here there are strong interactions here, strong interactions here. And so we can basically say that these are forming boundaries, like CTCF mediated boundaries or chromatin loops, right? And if we were to zoom in even further, we could now capture the nucleosome position around that protein binding site because we're doing an MNase-centric view. So it actually is a little bit of a misnomer. It combines the Chip-seg, the HiC, and an MNase positioning assay as well. And so what this really means is that you can dial in your topology analysis around features of interest, right? So here we're just looking at CTCF HiChIP data for at 100 million read pairs compared to a multiple restriction enzyme approach to HiC at 800 million read pairs. Again, metagene analysis around CTCF sites where this is just an average of many contact matrix squares. And so what we can see here is we see a nice bright punctate signal where CTCF are interacting with very little background noise. And again, with the restriction enzyme-based approach, you see that, yes, there is indeed a hotspot, but that's associated with a lot of background noise, which kind of makes it hard to identify these loops using different algorithms. And so we have a wide range of validated antibodies to do this. Here I'm just showing random snippets of H3K4-trimethyl, CTCF, H3K27. These are currently what we're offering with our services, and I'll show you our full list of antibodies that's supported by our kits. And one of the questions that we often get is, how is our HiChIP different from other HiChIP assays? And really, again, it fundamentally comes down to the use of restriction enzymes. So here, what I'm plotting is normal Chip-seg data for CTCF, HiChIP coverage for the Dovetail approach, and then HiChIP coverage for a restriction enzyme approach. And then I'm calling out where the restriction motifs are here at the bottom. And so really what restriction enzymes do is they distort the peak. So here we're able to recapitulate the peak signal. And in this case, it's subtle, but there's a dual, there's a pileup of restriction sites right here. There's 2 of them. And now we see that the peak is actually shifted over because you're enriching data on that restriction site. Here again, we see the peak, a peak that we can recapitulate using the MNase approach. And then with the restriction enzyme, there are restriction enzymes that flank this, this chip peak, but none that occur directly over it. So now what you get is this heavily distorted peak. And then in extreme cases, you completely miss the peak, where you're really only enriching data over nearby restriction sites and not over the feature of interest. And so again, using MNase is just more centric to the fundamentals of chromatin biology. So here is our full list of validated antibodies that you can pair with our HiChIP kit. And with that, I would like to thank you all for listening to me talk, and I will take your questions now.